If you've seen Jurassic Park and I asked you how to bring back a T-Rex, you probably think we need to stumble upon some DNA trapped in amber. But unfortunately, the DNA would have degraded long ago, even in the best conditions. The real key is solving a few completely separate, much weirder problems. Starting with the hardware, the genetic code is lost to time, so the only solution is recreation. To use what we know about them and taking clues from living relatives to feed into an AI system to write the code from scratch. To be clear, this and everything else we're going to be talking about are extremely difficult tasks. We won't be doing this next year, but this might be possible in our lifetimes. Okay, so even if you recreate the genetic code perfectly, it would die after its first meal. That leads us to another problem, the software, and the secret to solving that is the vulture. Then there's the issue of the eggs. We can start with small dinosaurs, but if we ever wanted to recreate the T-Rex, we need something that will lay massive 11-pound eggs. Humanity will need to build up the tools to make this possible, and that starts with the more recently extinct dodo. We have almost perfect copies of dodo's DNA, so this is a much easier problem than a T-Rex, and the way we resurrect a dodo is roughly the same as how we resurrect a dinosaur. But birds are still tricky. You can't directly swap nuclei in a yolky egg. For mammals, we can directly swap out the genetics of an egg and replace it with the new genetics of the animal that we want, like replacing mammoth DNA into an empty elephant egg. This is challenging enough. But with birds, there's an extra step. You have to take a living relative's primordial germ cells, aka PGCs, the cells that become sperm or eggs, instead of the eggs themselves. In the case of a dodo, you take a pigeon's PGCs, then rewrite the genetic code within them. You then inject those edited cells into a pigeon hatchling. Instead of creating a dodo directly, this creates a chimera. From the outside, it will look like a normal baby pigeon, but after it becomes an adult, it will have a dodo as its offspring. Do that to a male and female pair, and you have an almost perfect replica of a dodo in the 21st century. This isn't science fiction, and this is already in the works and might even happen as soon as 2028. After the dodo, the next step is going bigger. The Epeornis maximus, aka elephant bird, was the largest bird that ever lived. They were 10 feet tall and 2,200 pounds. That's 8 times heavier than an ostrich and the weight of a small car. The process will be roughly the same as the dodo, but it gets a bit trickier and will teach us more about the complications of resurrecting dinosaurs. Its closest living relative is the kiwi, but there's a small issue with this. The kiwi is 4 pounds. No way could a kiwi lay an elephant bird egg. So instead, we take the kiwi's PGCs, modify those with elephant bird genes, then transplant the modified kiwi PGCs into an ostrich embryo. So everything is the same so far, except that we need to use kiwi PGCs instead of elephant bird PGCs. But you might be thinking, isn't the egg still too large? And yeah, it is. So we will need to first genetically modify normal ostriches to increase their own size and hips so it can handle the 13-inch elephant bird egg. T-Rex eggs were roughly 17 inches, so we wouldn't be that far off at this point. After that, we need to make sure the egg stays alive until it hatches. For that, we need to engineer a macro incubator perfectly tuned for the elephant bird. Alright, so by now, researchers have figured out how to modify massive amounts of genes in PGCs, produced oversized previously extinct animals, and created perfectly tuned incubators. Now we can just do this with a dinosaur, right? Unfortunately, or fortunately, depending on the way you look at it, we can't. The genetic code is still lost. We'll need a sort of computational resurrection. You see, some pieces of DNA don't disappear when species change through generations. They're simply turned off. That means we can use the living dinosaur descendants, birds, which technically have dinosaur DNA living within them. So what we could possibly do is feed hundreds of modern bird genomes into AI systems that run ancestral sequence reconstruction. We'd also slip some crocodile DNA in, as even though they sit further back on the evolutionary tree, their DNA has remained remarkably stable over millions of years and could be useful for the system to figure out prehistoric genetics. Think of it like trying to reconstruct Latin by only looking at modern languages. You'd line up their dictionaries and find words that are similar, like the English father, the Italian and Spanish padre, and the French père. You'd infer they all came from a common root word, likely the Latin word for father, pater. This could get us to a sort of best guess on the species we're trying to target, but it would still have lots of gaps. But how do we know our best guess is any good? You may or may not have heard of the Nobel Prize winning system that is AlphaFold, the AI system that predicts protein's 3D structure, among some other things. It's gotten a ton better in predicting how biology works in its first few iterations, and they have no plans on slowing down. AlphaFold 2 was the best in class predictor of protein folding, the microstructures that power all of biology. AlphaFold 3 can predict certain interactions with proteins and other molecules. And they just want to keep working up from there, predicting larger and more holistic systems of biology. This and systems like it, at a base level, act as a biological spell checker. We could give it a reconstructed gene and ask, if this existed, would the protein it creates fold into a stable, functional shape? It could tell us if it produces a tangled mess, in which case we could go back and fix it. But for a challenge as large as reconstructing an entire dinosaur genome, we need more than that. Nobel Laureate Demis Hassabis wants to take AlphaFold from predicting proteins to predicting entire cells. This would allow us to truly understand the environment we would be creating for our rebirthed dino, making sure there's no unforeseen side effects. Even after all this still wouldn't mean we have a one-to-one reconstruction, but theoretically, we could get pretty close. So now say we've achieved this scientific marvel. We have an approximation of the DNA, but at this point, it still just lives in a computer and is not physical. Today, we can synthesize and print about 1 million DNA nucleotides. For almost any dinosaur, we would need to scale that up to billions. There are projects that aim to print a human-sized genome in the 2030s. If this is achieved, we are one step closer to Jurassic Park, whether you think that's a good thing or not. Then there's the extremely overlooked aspect of any animal's well-being, its gut microbiome. If we manage to do all of the above without thinking about this, the animal would die after its first meal. See, every animal isn't just one organism, it's an ecosystem. In fact, there are more bacteria living inside you right now than you have cells. Modern mammals get their gut bacteria required to live from the birth canal and breast milk. Modern birds and reptiles from the mom's preen glands in their tail and nest soil. A lab incubator would give them nothing. And the T-Rex's stomach acid and microbiome was most likely extremely tough, as it probably wouldn't always eat fresh meat. This is where the vulture comes in. Vulture stomach acid is at a pH of about 1, just slightly less acidic than battery acid, which means the bacteria that live in it is extremely tough. Botulism isn't even able to pass through. So here's the rough plan to kickstart a modern T-Rex's stomach. Collect and grow microbes from vultures and possibly Komodo dragons, both with incredibly tough and harsh gut ecosystems. Then create a so-called robogut, which is simply an acidic solution kept at about 100 degrees Fahrenheit, mixed with a whole bunch of microbes. This will be the competition space for microbes to battle it out and show who can work together. We slowly rotate the solution to mimic churning and intestinal flow and add low oxygen valves to create the same conditions that are in real guts. Then drop food scraps in over time that are similar to what a young T-Rex would eat. Over time, the fiercest and most cooperative strains outcompete the rest, forming a balanced community. Then we need to test the community with some living gut tissue to make sure it doesn't damage the cells. After that, we can freeze dry them into tiny capsules, which are basically probiotic pills. Feed the new hatchling once per week, and maybe, just maybe, we have done everything we need to recreate a living and thriving T-Rex. Thanks for watching.